Now we're going to look at the nitrogen cycle. There are a lot of elements that cycle in this earth, some of them more significant than others, and nitrogen is probably one of the most important ones. And there's certainly a lot of information about microorganisms being involved in that. So we're going to talk about that. We're going to start off with that the atmosphere is going to contain 70% nitrogen. Unfortunately, that nitrogen is not in a form that we can use. But nitrogen also makes up as much as 15% of your cell mass. We have nitrogen in our DNA, we have it in all of our proteins, and so a fair amount of it. But we're not able to use the nitrogen from the air. We have to get it from somewhere else. And what we see is that bacteria are the only organisms that we know of at this point in time that's able to take the nitrogen gas out of the air and put it into a form that can be used by all other living organisms. So that's what we're going to be talking about. The first step is going to be called nitrogen fixation, and I like to think of it, when nitrogen gas is in the air, it's broken. We can't work with it. So it has to be fixed by the bacteria. So in nitrogen fixation, that nitrogen gas is going to be converted into a compound that's called ammonium. So if you look at it chemically, we're basically adding hydrogens to the nitrogen. And I put the equation here into the NH3. This is obviously not chemically balanced whatsoever. It's just kind of a summary. Now, there's an enzyme in these microorganisms that's responsible for carrying out this process, and it's called nitrogenase. It's rather complex, which is kind of interesting. And it has to be to carry out this process that it's going to carry out. First of all, it's going to have to be able to bind to nitrogen. It's also going to have to bind to some compound that has hydrogen so that it can add the hydrogen to the nitrogen. And that's going to take a lot of energy as well to carry this out. What's interesting is this cannot happen in the presence of oxygen. That inhibits the enzyme so that it pretty much has to have an anaerobic environment. And there are a number of ways that these organisms achieve an anaerobic environment where the nitrogenase can be. So with nitrogen fixers, there are two different kinds. One of them is called free living, and the other one is going to be associated with plants. I'm going to start off talking about the free living ones, and as the name implies, they are living independently. And I'll show you an example here of some nitrogen fixers. But what happens is some of these organisms will form, or at least the free living ones will form this structure that we call heterocysts. And it's got a fairly thick wall that helps to keep oxygen out, so we end up with the anaerobic environment in these specialized cells known as heterocysts. Two examples of genera that are able to do that. Nostoc is one, Anabina is another. So when you see those names, free-living nitrogen fixers. We're glad to have them. If they're not free-living, we're going to find them associated with plants. And they have a symbiotic, and I've clarified that with mutualistic relationship. So symbiosis is a term that refers to a relationship between two organisms. So you can have a relationship where both of them benefit, one of them benefits, or one benefits, there's harm. Those are all symbiosis. But we tend to use symbiotic, and we really mean mutualistic, where both of the organisms are going to benefit. So when we have this association between the nitrogen fixers and plants, that's going to be a case in which both of them are going to benefit. So the plant will end up with a little nodule on them that creates an anaerobic environment, which is what the nitrogen fixer needs. And then the nitrogen fixer itself, the bacteria, is going to be providing nitrogen for the plant. An example genus of this is Rhizobium. And I have a picture of a plant root, and you can see these little nodules on here. Those are the nodules that the plant has formed with the rhizobium inside them in that anaerobic environment. And here they have a more close-up view of it, or sort of like a cross-section. You can see one of the nodules, and there's showing you where the rhizobium is. and where the central nitrogen fixing zone is. So that's where most of the nitrogen fixation is going on. And then that once the nitrogen is changed to ammonia, it is leached out into the plant root, and the plant uses it. Here's a good example of a root system that's got some relatively large nodules in it. Now, if you dig up clover or pea plants that have not been fertilized, there's a good chance that you will see these, especially if they've been there for a while. So, with the nitrogen cycle, we have the atmospheric nitrogen, and it diffuses down into the soil where the nitrogen-fixing bacteria get hold of it. The ones that are associated with plants are going to feed it in the ammonia directly into the plant. The free-living ones are going to take this nitrogen gas and make it into ammonium, like I showed on the earlier slides. Now, we can also get ammonia from degradation of plants, and probably animals too, as they die. Decomposers, as we show here, aerobic and anaerobic bacteria and fungi are going to start breaking it down, and then they release that process into the soil. And that process is known as ammonification. And in ammonification, organic nitrogen, which means it's nitrogen that's already attached to material from a living organism or formerly living organism, and then that can be used by, again, plants, or go further into the processing of the nitrogen. So with the ammonia, it can be used directly by other plants and microbes and animals, especially by animals that are eating plant material. Or another group of bacteria called ammonia oxidizing bacteria get hold of it, and they are going to oxidize it into a different kind of compound. And so we're showing you that right here we have the process called nitrification, where the ammonia is converted to something called nitrites, which are NO2. And of course, they're called the nitrifying bacteria. So in this process, we're going to get nitrate. It actually is a two-step process to get to nitrate. We have to get to nitrite first, and those ammonia oxidizers I was talking about take that ammonia and convert it to the nitrite, and then that's followed by another group of bacteria called nitrite oxidizers that take the nitrite and convert it into nitrate. So it is a two-step process requiring two different populations of microorganisms. Now, these are considered to be chemoautotrophs. They're getting their energy from chemical compounds, but they are getting their carbon from a non-living source. In the particular case with the chemo, they're getting their energy from nitrogen. And here I'm showing you the pathway, and they're showing you in this image here how the ammonia goes in. It gets converted by the ammonium oxidizing bacteria to nitrite, and then the nitrite oxidizing bacteria give you the nitrate. So we have this nitrate, and what can we do with that? So it can be used by plants and microorganisms in the process to make DNA and protein. We call that assimilation. Why did I put the picture of this that I did here? If any of you are old Star Trek fans and you know about the Borg, they assimilate other species, so I thought that was a good tie-in. Nitrates can also get into our waterways, and they cause this process that we term eutrophication. And what happens is you get enough nutrients in the water that plant life and algae and that sort of thing in the water grows really, really dense. It can even make a covering or a layer over the water so that oxygen's not able to diffuse down in there. And then the animal life like fish, don't have enough oxygen in the water for them to keep themselves going, and they end up dying, and you end up with a big smelly mess. Another process that takes place is going to be called denitrification, and in this process, the nitrate is going to be converted back to nitrogen gas. So of all these that I have listed here, assimilation is what we would really like to see most happening, because that's how we're going to get our nitrogen, is because we're going to be eating plants and microbes, so we can get the nitrogen from them to use in our DNA and protein. I threw in this little slide here, or this little image here, where it talks about drinking water systems that violate the EPA health standards. I don't know what year this came from, but it's an indication that probably there's too much nitrogen in the drinking water. But it shows you that we're in Oregon, we're not in the best category for some reason. So what we've talked about then was this last step, was the denitrifying bacteria taking the nitrates and converting it back to nitrogen. So you can see how everything is getting cycled here, and we'll come back to that a little bit. Here we have both bacteria and fungi converting the nitrate to nitrogen gas, and just like the very first step where we convert nitrogen gas to ammonia, it occurs under anaerobic conditions. And this can happen in wetlands and riparian areas. It intentionally happens at wastewater treatment plants, where we're having the biological waste being dealt with by the various microorganisms. And somebody termed this as being one of the least beneficial aspects of the nitrogen cycle, because it is putting the nitrogen back as nitrogen gas, and that means we're not able to use it. But then it can continue the cycle that we've been talking about. So, looking at it from a slightly different standpoint, here we have the N2 that's going to go through the process of nitrogen fixation, and then we have this CnH2 that represents amino acids or organic forms of nitrogen, and it goes through the process of ammonification to give us the NH3. And then from the NH3 we have the nitrite oxidizers, ammonia oxidizers to give you NO2, and the nitrite oxidizers to give you NO3, and that's the process of nitrification. And then the nitrates can either be assimilated, which is what we really want to see happen, and when that happens we're back to here where we have the amino acids and the DNA, or we go through the step of denitrification where we produce the nitrogen gas again. So this is kind of the cycle drawn out a little bit differently so you can see.